Hey guys, I wanted to talk to you today about viral replication. Some people refer to it as multiplication. We know that viruses are not alive, so instead of living and dead, we use things like active and inactive. So sometimes people use the word multiplication when they're talking about a virus being able to copy itself. All right, so let's go to the PowerPoint. And a virus is defined as an obligate intracellular parasite. So parasite knows that it needs to be have a host and it causes damage to that host. Intracellular, it has to be within a host cell to cause this damage. Obligate, that's, it's obligated to be there. A virus can't replicate by itself. It has to have a host. All right, and it does this by taking over the host machinery. It uses our ribosomes, it uses our enzymes, it uses the host's ability to replicate itself, and it uses those tools in order to replicate a virus. All right, so why do we care about how it does this? Well, viruses can be very individual. They can all do this slightly different ways. We can put them in groups according to how they do that. But when we know how it replicates itself, we can find out things of interest to us, like how the virus is transmitted, what damage it causes to the host, and how our immune system responds to it, because sometimes our immune system is a factor in the damage caused by viruses. And then ultimately, our main goal is to control this viral infection in a host. Okay, so let's look at a way the virus can multiply. So phases, we can separate this multiplication cycle into 5 different phases. Those are adsorption, penetration, which includes uncoating, synthesis, assembly, and release. Now this cycle can vary depending on the virus. Some viruses do this very quickly, maybe 8 hours. Some viruses take a very long time. What's the difference? Well, it depends on from the time that it invades a host, how long are you going to get enough virions in your system and cause enough damage to show symptoms. So that makes a difference too, because we know you can be asymptomatic at the beginning of a virus. We're learning so much about viruses with the SARS-CoV-2 right now. Okay, so adsorption. Let's look at each one of these in detail. The word adsorption, really we're talking about attachment here. Adsorption, and it really is a D, it's not absorption, which is a liquids, it's adsorption. It's a chemistry term, and basically we're just talking about attachment. So how does this happen? The very first thing is you need a virion to attach to a host cell. How does it do that? Well, remember in the structure, we talked about those spikes, those glycoproteins that are on the surface of a virion. These glycoproteins, these spikes or glycoprotein spikes will attach to a protein on the host cell. Now, this protein on the host cell, this is just a regular protein. This is a protein that the cell is using to do cell stuff. It's just a regular protein. However, this particular virus has a complementary protein that will attach to it, and it allows this virus to get inside. Beneficial for the virus, not so much for the host. Okay, so this has to happen first, this stage of absorption, which is attachment, which is involved protein-protein interactions. Okay, so depending on these proteins depends on what type of cell this virus can attach to. If the virus cannot attach, the virus can't get inside the cell, the virus can't replicate, no harm is done. So we are only worried about the viruses that can attach to the host. All right, we refer to that as the host range of the virus. And different virions, different viruses have different host ranges. Some are very specific. In fact, the human virus hepatitis B, it will only attach to liver cells in humans. It doesn't attach to other cells, muscle cells, nerve cells. It doesn't attach to other animals. Other mammals don't get hepatitis B. So it is very specific and limited host range. Others have a little more moderate. Polioviruses can attach to intestinal cells and nerve cells, which means the damage is done. Back to hepatitis, if you know a little bit about the disease hepatitis, you know that the signs and symptoms, the damage that it causes are only to your liver. Poliovirus can attack intestinal cells and nerve cells, so you get damage in those. It also attacks primates, so it's not just humans that can get polio, but our but other animals, other primate animals as well. Rabies, we know what that is. Rabies, you can get that by a bite of a rabid dog or a skunk or a bat can carry rabies. All mammal cells, all mammals, there's an exception. I think there's one, I think possums or something can't carry rabies. There's some exceptions, but mammals have the same protein that the rabies virus can attach to, which means it can attach to all different types of mammal cells. Okay, if we don't have this protein, then we are naturally immune from the virus. This is why we can't give COVID to our dogs. Our dogs do not have the same protein that the COVID SARS-CoV-2 virus uses to attach to our cells. So they are naturally immune. We are naturally immune to a lot of different viruses, a lot of different things that only infect plants, only infect other animals. So that is a natural immunity there. Okay, so that is the first step. The first step to a viral infection and viral multiplication is this attachment, which we refer to as adsorption. Okay, so now we've got it attached. Now what happens? Now it needs to get in. It needs to penetrate. So that is stage 2. Attachment has occurred, and now this virion needs to get inside a cell. Now, in animal viruses, in viruses that affect humans and other animals, there are 2 ways that these virions can get in the cell. One is by endocytosis, which you should know what that word is, so that's very familiar, and another one is membrane fusion. Fusion, if you talk about something, if you're going to fuse something, you need 2 things, a minimum, to fuse together. These are membrane fusions, so we need 2 membranes, right? The first membrane is going to be our cell membrane on our host cell. The second membrane is going to be the virus. Which viruses have membranes? Well, those are going to be those enveloped viruses. The virus that has an envelope has a membrane, a phospholipid bilayer from that. So membrane fusion only happens for enveloped viruses. So that is a requisite for membrane fusion. This only occurs for enveloped viruses. Endocytosis can happen for either naked viruses or membrane enveloped viruses can enter by endocytosis. If a virus enters by membrane fusion, it will always enter by membrane fusion. So it has the genetic instructions in the viral DNA that it will always fuse membranes. So whichever way a virus does it, it will always do that. Now, another vocabulary term is uncoating. Once it gets inside, it needs to, the nucleic acid needs to be released from the capsid, and that is referred to as uncoating. All right, let me go to the picture because pictures are better. These are the 2 different types. Here we see endocytosis. So you can see the virion. We see absorption occurring with that attachment going in here. That will trigger the cell to engulf it. So endocytosis, it comes in a vacuole surrounded by that. And then there are enzymes inside the cell or in the virus that will break down that vacuole and then release the viral nucleic acid in there. So that's the endocytosis. The other type is that membrane fusion. Okay, so here you see some attachment all along the membranes, and that starts a different chemical reaction, causes the membranes to fuse to one another, and then the membranes to that envelope virus gets integrated into the host cell membrane, and then that allows the capsid, the nucleocapsid, the capsid with the nuclear material inside to come inside, and then encoding happens within the cytoplasm of the host cell. Okay, so that is the first 2 steps for viral multiplication. We have adsorption and we have penetration. Okay, that's the start. We'll do the next video and do the rest.